This podcast is an overview of the unique functions of the liver and the pathways that either occur primarily in the liver or mostly in the liver. Additional podcasts will likely be posted on the individual pathways that the liver is uniquely responsible for. There are 5 distinct functions of the liver that are unique to the liver or are mostly occurring in the liver versus other tissues. The first unique function of the liver is its ability to restore blood glucose during the fasting state to prevent hypoglycemia. This key liver function involves 2 pathways, glycogenolysis and gluconeogenesis. During fasting, those 2 processes are coordinated to produce glucose, release it to the bloodstream, and maintain glucose levels in the plasma so that other tissues can use it. So in that sense, the liver is an altruistic organ looking after the other tissues that need glucose for their own survival. There are 2 important tissues that specifically depend on glucose in the bloodstream, and those are the red blood cells and the brain. The second unique function of the liver is the biosynthesis of cholesterol. The liver is the major organ that synthesizes cholesterol. As such, the liver provides other tissues with cholesterol for membrane synthesis, vitamin D synthesis, and steroid hormone synthesis, and it uses cholesterol for its own metabolism as well as the synthesis of bile salts. There is limited cholesterol biosynthesis in several other organs, including mammary glands, reproductive organs, and adrenal gland. The third unique function of the liver is the synthesis of ketone bodies. During fasting, ketogenesis is increased due to the accumulation of Acetyl Coa in the liver, and the liver is uniquely situated to where it has all the enzymes required for the synthesis of ketone bodies from Acetyl Coa. The ketone bodies thus synthesized in the liver are released in the bloodstream to provide peripheral tissues with alternative energy sources when blood glucose levels are low. Importantly, ketone bodies produced by the liver cannot be consumed by the liver. So the liver is the only tissue that can produce ketone bodies, yet it cannot use them for its own energy needs. The fourth unique function of the liver is the urea cycle. In the urea cycle, the liver is able to dispose of nitrogens and ammonia derived from the catabolism of amino acids, and as such it detoxifies any ammonia produced in the liver or peripherally or collected from peripheral tissue, and by producing urea it can package those nitrogens into a molecule that is transportable and easily excretable. The fifth unique function of the liver is its ability to detoxify organic molecules, and this is primarily done through the NADPH dependent monooxygenase system, which can make organic molecules more water soluble and allow their excretion through the kidney. This overview is not an all-inclusive list of all the unique processes that occur in the liver. The liver as a glucose factory. One of the unique functions of the liver is its ability to produce glucose during fasting. The glucose released by the liver into the bloodstream restores blood glucose and allows various cells and tissues to access the energy source they need the most. Glucose production in the liver is a coordinated process between glycogenolysis and gluconeogenesis. Glycogenolysis breaks down glycogen, providing glucose from glycogen stores, whereas gluconeogenesis will use smaller molecules as precursors like pyruvate, lactate, glycerol, and other carbon skeletons derived from amino acids to produce glucose. Both of these pathways, glycogenolysis and gluconeogenesis, are more active in the fasting state. In general, these two pathways are activated by a rise in glucagon and or epinephrine in the bloodstream, which will result in the activation of specific enzymes in those two pathways. The key enzyme that breaks down glycogen is glycogen phosphorylase. This enzyme is activated by the actions of glucagon on the glucagon receptor or epinephrine on the beta adrenergic receptor, causing a rise in cyclic AMP, which in turn activates pKa. pKa can phosphorylate glycogen phosphorylate kinase, and glycogen phosphorylate kinase can then phosphorylate glycogen phosphorylase and activate that enzyme. The outcome of activating glycogen phosphorylase by phosphorylation is to break down glycogen and eventually produce glucose that is released from the liver into the bloodstream. Alongside glycogenolysis, gluconeogenesis will also begin during the fasting state, and that process is also activated by glucagon. Gluconeogenesis will use carbon skeletons derived from several amino acids, including the carbon skeletons pyruvate, oxaloacetate, alpha-ketoglutarate, succinyl CoA, to make glucose from these smaller molecules, with lactate and glycerol providing additional sources for the synthesis of glucose. Glucagon activates gluconeogenesis by its effects on three key enzymes. These key enzymes are PEP carboxykinase, fructose 1,6-bisphosphatase, and glucose 6-phosphate phosphatase. All three enzymes are upregulated, or the expression of all these three enzymes is increased by glucagon. So gluconeogenesis becomes more active when glucagon is elevated in the plasma. Another important allosteric regulator of gluconeogenesis is acetyl CoA. Acetyl CoA in the liver will accumulate due to beta oxidation of fatty acids by the liver, using the former molecules as a source of energy. Acetyl CoA thus produced will activate the first bypass enzyme in gluconeogenesis, pyruvate carboxylase. The common molecule produced from gluconeogenesis and glycogenolysis is glucose 6-phosphate. The liver is unique in that it has glucose 6-phosphatase, which will produce glucose from glucose 6-phosphate. Without this former enzyme, glucose would not be produced from glucose 6-phosphate and could result in severe fasting hypoglycemia. The second unique function of the liver is that it is the major site for cholesterol synthesis, starting with acetyl CoA. The liver can ultimately determine how much cholesterol is produced in the body. The outcome of cholesterol production by the liver will have two other important effects. The first one is that the liver output of cholesterol will ultimately determine the plasma levels of cholesterol. And the second one is that the liver can serve as a major storage site or depot for cholesterol by its ability to convert cholesterol to cholesterol esters and store it in specialized lipid vesicles. Cholesterol biosynthesis in the liver is tightly regulated and is dependent on the rate-limiting enzyme in the liver, hydroxymethylglutaryl-CoA reductase, or HMG-CoA reductase. There are several ways where HMG-CoA reductase activity is regulated. The first one is that cholesterol can inhibit this enzyme directly as a feedback inhibitor. Also, mevalonic acid, which is the product of HMG-CoA reductase, can inhibit this enzyme competitively as a feedback inhibitor. A second way cholesterol can regulate the activity of HMG-CoA reductase is that the concentration of cytosolic cholesterol inside hepatocytes can regulate the gene expression of HMG-CoA reductase in the liver. High concentrations of intracellular cholesterol will downregulate the gene expression of HMG-CoA reductase, and conversely, low intracellular cholesterol will upregulate the gene expression of HMG-CoA reductase. A third regulatory mechanism of HMG-CoA reductase in the liver involves the hormones insulin and glucagon. Insulin can promote the dephosphorylation of HMG-CoA reductase through stimulating a phosphoprotein phosphatase. The outcome of this covalent modification is to activate the enzyme. On the other hand, glucagon can promote the phosphorylation of HMG-CoA reductase and consequently inhibit the enzyme. HMG-CoA reductase can be specifically covalently modified or phosphorylated by the protein kinase AMP kinase, and that also results in the inhibition of the enzyme. In addition to cholesterol being able to regulate the gene expression of HMG-CoA reductase, high intracellular cholesterol can regulate the degradation of HMG-CoA reductase in hepatocytes. High intracellular cholesterol concentrations will promote the ubiquitination of HMG-CoA reductase, which is an important signal to send HMG-CoA reductase to the proteasome, where it will be degraded, and as such it will be removed from the cytoplasm of hepatocytes, resulting in decreased cholesterol biosynthesis. As mentioned before, ultimately the amount of free intracellular cholesterol will determine the amount of plasma cholesterol. Low intracellular cholesterol will increase the gene expression of the LDL receptor on the surface of hepatocytes. When there are more LDL receptors on the surface of hepatocytes, more LDL particles can be endocytosed from the plasma, thus reducing their concentration and eventually reducing the total amount of plasma cholesterol. In fact, this is the therapeutic strategy behind the use of statins to reduce plasma cholesterol. The inhibition of HMG-CoA reductase by statin results in a decrease in intracellular cholesterol concentration, which in turn results in an increase in the gene expression of the LDL receptor. More LDL receptors on the surface of hepatocytes will endocytose more LDL from plasma and thus reduce plasma cholesterol. The third important function of the liver in terms of cholesterol storage is that it has an enzyme abbreviated ACAT, Acyl CoA cholesterol acyltransferase, which is the enzyme responsible for converting free cholesterol into cholesterol esters. Cholesterol esters, once formed, can be packaged and stored in the liver. By esterifying cholesterol and packaging it in special storage vesicles, ACAT can contribute to the reduction of free intracellular cholesterol within the liver. Stored cholesterol esters in the liver can be later used for the liver's own metabolism, such as the synthesis of bile salts. The third unique function of the liver is its ability to produce ketone bodies. Ketone bodies are synthesized from acetyl CoA by condensing two molecules of acetyl CoA first by the enzyme thiolase. A third molecule of acetyl CoA is added to acetyl CoA to form HMG-CoA by the mitochondrial HMG-CoA synthetase, to be distinguished from the cytosolic HMG-CoA synthetase involved in cholesterol biosynthesis. HMG-CoA is further metabolized by HMG-CoA lyase, which will produce the first ketone bodies, acetoacetate. Acetoacetate can be further metabolized to produce acetone spontaneously or beta-hydroxybutyrate enzymatically. So the two major ketone bodies produced by the liver and found in circulation are acetoacetate and beta-hydroxybutyrate. The ketone bodies acetoacetate and beta-hydroxybutyrate released by the liver will provide an alternative energy source to multiple tissues, including the brain, muscle, and other peripheral tissues. It is noteworthy that ketone bodies are an important energy source for many peripheral tissues during fasting. It's important really to just appreciate the big picture about ketone bodies and how they're used and why they're important. So ketone bodies are derived from acetyl CoA, and in turn that acetyl CoA comes from the beta oxidation of fatty acids in the liver. So the liver will use the beta oxidation and acetyl CoA for its own energy needs. At some point there'll be enough energy, ATP and NADH, that the TCA cycle gets shut down, so you have an accumulation of acetyl CoA. And as a result, acetyl CoA gets shunted into making ketone bodies in the liver. So that's the first important thing. is that the only organ capable of making ketone bodies is the liver. The second important thing is, why do you need ketone bodies? Acetyl Coa that accumulates in the liver cannot be released. You have to remember the coenzyme A molecule is huge. That cannot be exported out of the cell. So by making ketone bodies, you're creating two new molecules, beta hydroxybutyrate and acetoacetate, that can be exported, released into the bloodstream. Okay, so that's the advantage of making ketone bodies, because you created a molecule that can be released into the bloodstream that can be used by multiple tissues, with the exception of the liver. The liver cannot use ketone bodies. It can make them, but it cannot use them. So once released in the bloodstream, ketone bodies can be picked up by a variety of tissues. Okay? Muscle, heart, even the brain. That's what's so important. The brain can pick up ketone bodies from the bloodstream when they reach a certain concentration in the blood. So that's the important thing. So knowing what they're used for, what's the advantage of having these molecules made, is that they can be transportable in the bloodstream and they can be used as an alternative energy source by a variety of cells, including the brain. Okay. The fourth unique function of the liver is that it has the urea cycle. And the function of the urea cycle is to detoxify any ammonia produced during metabolism of nitrogen-containing amino acids and other nitrogen-containing molecules. So let me say a couple of things about the fate of the ammonia we produced and how we're going to dispose of it. Okay, so I'll just give you an overview and I'll stop and continue tomorrow. But basically, I want you to appreciate that if we leave that ammonia in the tissues, it is very toxic. So we have to get rid of it fairly quickly and in a non-toxic way. Hence the need of a non-toxic disposal of that ammonia. Okay? The liver has a very important cycle. The only tissue that has a very important cycle, the urea cycle. The function of this cycle is to take ammonia or nitrogen derived from amino acid degradation, incorporate those nitrogens into a non-toxic molecule called urea. The urea molecule contains 1 carbon, 1 oxygen, and 2 nitrogens, or 2 amine groups. So that becomes, when it's made, that is a non-toxic molecule. It can be released in the bloodstream. It can, that urea is carried to the kidney, and it can be disposed of safely. Actually, even through the blood is non-toxic. Okay, so that's really the importance of the urea cycle to dispose of nitrogen derived from degradation of amino acids. Occurs only in the liver, and it's a non-toxic packaging, if you want, of amines derived from catabolism of amino acids. I just want to show you here in the urine, nearly 90% of all nitrogen in the urine is in the form of urea. And it is coming from the degradation of amino acids. It's carrying the nitrogens that from degraded amino acids. Okay, there's other nitrogens, but urea is the major way we get rid of nitrogen after we degrade amino acids. Okay, I'll stop here. The fifth unique function of the liver is its ability to detoxify organic molecules to enhance their disposal from the kidney. In fact, there are two such pathways in the liver that are involved in the conversion of organic molecules into more soluble forms that can be more readily excreted. The first of such pathways is the Microsomal NADPH Cytochrome P450 dependent monooxygenase system in the liver. This monooxygenase system in the liver is associated with the endoplasmic reticulum, and it specifically functions to introduce oxygen molecules into organic molecules to make them more water soluble. As a reminder, the NADPH required for this monooxygenase system is primarily derived from the pentose phosphate pathway or the glucose 6-phosphate dehydrogenase reaction that produces NADPH. An alternative source of NADPH is the malic enzyme, which can break down malate to pyruvate and CO2 and also produce NADPH. It is also noteworthy that the liver has a separate monooxygenase system that is associated with the mitochondria, and that monooxygenase system is primarily involved in the metabolism of steroid hormones. The second important detoxification mechanism involves the addition of glucuronic acid to organic molecules to again make them more water soluble. This reaction is catalyzed by UDP glucuronyl transferase, which uses UDP glucuronic acid as the donor of the glucuronic group. This conjugation of glucuronic acid can use a variety of organic molecules as substrates, but in particular, bilirubin is a key substrate for the addition of two molecules of glucuronic acid through UDP glucuronyl transferase, and thus this conjugation will enhance the disposal of bilirubin from the liver.